DNA Replication by Kinza and Eaman. Two methods were suggested on how DNA replication could occur. One method is the semi-conservative method, where the DNA is split into two and then it's replicated. The second method is the conservative method, where the parental DNA is not split and it is copied as a whole. Scientists later find out that the semi-conservative method is actually what is being used in DNA replication. There is actually a specific area where DNA replication begins, known as the replication origin. This area has a certain DNA sequence that becomes the starting point of the replication. Since the DNA is so long, there are many replication origins present throughout the DNA. The first step of DNA replication is unzipping the zipper, where helicase, an enzyme, is a zip and DNA is a zipper. Helicase goes through the DNA and breaks the bonds between the complementary base pairs. As you can see, the helix is now opened and the top strand goes from 5' to 3', bottom goes 3' to 5', making both the strands anti-parallel to each other. Since the bonds of the complementary base pairs are really strong, single-strand binding proteins are put on both the strands to prevent them from coming back together, and DNA always replicates from 5' to 3'. RNA primase creates a little segment known as the RNA primer that attaches to the DNA so that the DNA polymerase III that creates the DNA knows where to start creating the new DNA. DNA polymerase is now putting the complementary base pairs and creating the new DNA. The top strand is able to continue replicating from 5' to 3' and needs no other RNA primer to help it start again and keep replicating in that direction. But for the bottom strand, it needs more RNA primers to be placed again and again. For example, if the helicase had only opened the DNA up till here, the replication will be going from 5' to 3' after the RNA primer. When helicase has opened the DNA strand even more, another RNA primer will need to be placed in order to be able to replicate anti-parallel to the template strand from 5' to 3'. You can only replicate towards the right side from 5' to 3' in this diagram, so you're going to need a new RNA primer every time helicase has separated the DNA even more. This creates different fragments from one RNA to the next. These fragments are called the Okazaki fragments. The need of the RNA primase again and again causes this process to be slow and therefore creates the slow and lagging strand. Up until now, DNA polymerase III was used. Now, DNA polymerase I comes and switches RNA primers with DNA nucleotides. After that, an enzyme called ligase joins the Okazaki fragments together and makes sure that the DNA backbone is bonded together correctly. DNA polymerase I also proofreads the nucleotides created by DNA polymerase III. DNA polymerase III may make mistakes while creating a complementary nucleotide. When a mistake is found, DNA polymerase II goes back and then replaces and fixes the mistake. The end result of the DNA replication are two identical double helix strands, and the nucleotides that were present were adenine, thymine, cytosine, and guanine, which are the complementary base pairs.